Welcome NEET Cognition team, future doctors. So how was your day? The first thing I'm going to ask you is whether you all took a relaxation time today. It's a weekend day. Definitely you need a relaxation. So because all the 6 days you must be going to school, catching up with your tuition and backlogs. And this Sunday you have to definitely have a relaxation time. If you have not started, allot 8 to 9, okay, for your relaxation. So today I'm going to start up with the warm up, okay, because see, all the athletes, have you seen the athletes what they do before they start doing the physical training? They have a mental warm up. For everything you need a mental warm up. Like, even suppose someone comes to your house, you prepare a dinner or a breakfast, okay, lunch, whatever it is. You have a plan, like you have a goal, like if they come, I am going to do this. So before doing anything, we plan and fix a goal, and then we try to execute it. So today what I wish to say is, before you start studying every day, remember your goal. This is a mental warm up, okay? As soon as you sit down, don't start studying something like opening the physics or chemistry, start studying. Don't do that. Open your book, close your eyes, fix your goal. Once you have fixed it, try to remember it daily before your study session, okay, before your deep work or before your live classes. You study on your own, whatever it is. You have to close your eyes and think of your goal. Under goal, I am going to divide it into long term goal and short term goal. What's your long term goal? To clear NEET. In your point of view, I am going to say, okay, to clear NEET. Wear the white coat. Join Medico. Okay, this is non-negotiable. Okay, this is your destination. This is your destination. Okay, to reach this long-term goal, you should have a short-term goal. What is the short-term goal? That is taking each step every day, which means being consistent of even half an hour study, focused study. I already told you about focus time. So focused study. So under goal, you have long-term goal and short-term goal. Your long-term goal is to be a medico. That is your destination, which is non-negotiable. So what you are going to do to attain that long-term goal is based on your short-term goal. That is right in front of us. We have half an hour live. This is the short-term goal. This goal is going to be only for 30 minutes. What is the goal? Going to fix your focus for just 30 minutes. So 30 minutes into 30 days. How many hours? Calculate and tell me. Type it in the comment section. Okay, now another point. So this is your goal. Most of the time, why do you feel anxious about the goal? Why do you feel anxious about the goal? Because you think goal as a rigid box. Goal as a rigid box. Okay, but what I wish to say is, fix your goal, but be flexible in attaining the goal. Be flexible in your pathway in attaining the goal. That is goal A, which you planned straight. Okay, if suppose that goal doesn't work, we are not going to leave the goal. We are going to attain the goal. But now we are not going to take this route. We are going to plan B, take some diversion, reach the goal. Even if that doesn't happen, are we going to drop the goal? No. We are not going to give up. Again, we are going to modify the path, see the dips, again peaks, then reach the goal. So we all think these things are waste of time. Actually, this is your transformation time to become that person. Transformation time to become that person who you wish. Okay, so today I shared you the mental hack, the first one as a warm up. Now I want you to fix your goal, close your eyes, fix your goal. We are going to concentrate for this 30 minutes, which is our short term goal. Okay, shall we start, students? See, this is past 6 days synopsis, okay, from the day 1 to day 6. So even if you have missed today, we are going to do it as a rapid fire session. Okay, these are the code words. I have mixed it up. I have not followed the exact NCERT pattern, because in the NEET exam what you see mix up of questions. So to feel that effect when you are studying itself, you have to do that. You have to get the practice. Okay, so first we'll start. The block 1 is day 1. We are not going to write up because we have all these informations will catch us up for 30 minutes. So first we'll start with the Robert Hooke. Robert Hooke, who is the one who discovered the cell, but he found out a dead cell because he used a dead plant tissue. Cork is a dead plant tissue. So inside the cell, he didn't see cytoplasm. What he visualized is only the plant cell wall. So he is the one who discovered cell wall. Now Robert Hooke discovered a cell which is a dead cell. He used the tissue, a dead plant tissue, that is the cork, and he is the one who discovered cell wall. Second point, Anton von Leeuwenhoek is the one who first discovered a living cell. Okay, he first discovered the living cell. This is very important. Now I am adding up the NEET TRAP wherever I can. Okay, so swapping both of the above. Suppose if they confuse you with the living cell, who first discovered the living cell or who first discovered a cell, you have to differentiate that he is the one discovered dead cell, but Anton von Leeuwenhoek discovered a living cell. Now Robert Brown, already we saw N for nucleus. Robert Brown discovered nucleus. Next is, this is a straight question. You can get this question in the match the following. This discovery is important because only after the discovery of nucleus there are a lot of transformation in the field of bioengineering. Okay. Next is Schleiden and Schwann. Who proposed the cell theory? Who formulated cell theory? Schleiden and Schwann. Then the fifth point is Rudolf Virchow, who said omnicellular is cellular, that is all new cells arise from the pre-existing old cells. Okay, he spoke about cell division. Now what is the trap here between Rudolf Virchow and Schleiden and Schwann? Here they can ask you that, see, I have mentioned here the incomplete trap versus the finisher. Who finished the cell theory? It is Rudolf Virchow. Okay, because they failed to explain how new cells are formed. Who failed to explain? Schleiden and Schwann failed to explain how new cells are formed. So Rudolf Virchow is the one who completed the cell theory, that is omnicellular is cellular, which means all the new cells arise from the pre-existing old cells. Okay, and he is the one who spoke about cell division. Next is the dimension and size chart. Mycoplasma 0.3 micrometer is the length, which is the smallest of the microorganisms, and bacteria 3 to 5 micrometer. Human RBCs 7 micrometer in diameter. These are important: length, diameter. The largest isolated single cell is seen in ostrich. It is the ostrich cell. Next longest cell is the nerve cell. Now micrometer, that is, it can be called as micron or micrometer. This is 10 to power of minus 6 meters. Here it is 1 in millionth. It is 1 in millionth. Are you able to recollect what we studied for the past 6 days, students? If you know the answer before, type it in the comment section that you are able to retain it. Now the trap is micrometer versus millimeter. For example, if you have to look here, 3 to 5 you will be knowing the answer, okay, 3 to 5, but micrometer, or they can write like this, they can give like, okay, millimeters. So now you have to clearly differentiate this. In the exam panic we may miss, so even a small thing matters for NEET because your goal is very high, right? Next is prokaryote versus eukaryote. Block 2 is my day 2 revision. We are going to see the missing structures, that is lack of membrane bound organelles. Can you name some of the organelles you have in the membrane bound organelles? Organelles are endoplasmic reticulum, Golgi bodies, mitochondria. All these are absent because prokaryote is it leads a simple lifestyle. No rooms inside that. But there is one organelle which is seen both in prokaryote and eukaryote. You call it as a universal organelle. Can you name? If you know the answer, quickly type it in the comment section. Universal organelle. What is the universal organelle? Do you know the answer? Quickly type it. What is the answer? Ribosome. Because they are the ones who produce the proteins for the cell. Lack of membrane bound organelles is the characteristic of prokaryote. You have membrane bound organelles only in an eukaryotic cell. Okay. Next is mesosomes. Next you have mesosomes. That is, they are the infoldings of the plasma membrane. Infoldings we saw on that day, they are infolding means like this, folds towards the inner side of the cell. Now mesosomes, what is the trap they can give here? Mesosomes, if they ask you mesosomes or organelles, are they organelles? No, they are membranous structures. They are membranous structures. Mesosomes are not organelles, isn't it? Now only we saw prokaryotes does not have any membrane bound organelles. Okay. So mesosomes are not organelles. They are just a surface infolding of the plasma membrane. I am going to ask you one question. Okay. In prokaryotic cells you have mesosomes. In eukaryotic cell also you have an organelle starting with word M. Both has a common function. What is that function? Respiration. Now quickly type the answer for this M. Eukaryotic cell which has an organelle starting with the letter M and which does the function of respiration. Same way mesosomes also has one of the function, that is respiration. Okay. Next is inclusion bodies. These inclusion bodies are seen in prokaryotic cell. They are non-membrane bound and they are the reserve materials of the prokaryotic cell. Examples of prokaryotic organisms. What are the examples of prokaryotic organisms? Bacteria, blue-green algae, Mycoplasma and PPLO. Under bacteria, what is the importance of bacteria? Bacteria are the most abundant and primary member of this prokaryotic organisms. Next you have blue-green algae. It is also called as cyanobacteria. Okay, but here you have a trap. You can mistake it as an algae and tick the answer eukaryotic plant. Okay, but you should remember it is a cyanobacteria is a prokaryotic organism, that is a bacteria. Next you have Mycoplasma. What is the importance of Mycoplasma? Quickly type the answer in the comment section. They are the prokaryote organisms which doesn't have cell wall. They lack cell wall. Then the second answer, if you know the answer, quickly type it in the comment section. What is that? It can survive in complete absence of oxygen. Complete absence of oxygen. Okay. Next, what is the expansion of PPLO? Pleuropneumonia like organisms. Okay, these are the examples of prokaryotic organisms and the importance of prokaryotic organism also we finished off. Next is block 3. This is the day 3 synopsis. Now tell me the layers, cell envelope layers. Okay, what are the cell envelope layers? Now, what are the cell envelope layers? Block 3. You have, can you name the 3 cell envelope layers? If you know the answer, type it, leave the answer in the comment section. That is, it is a chemically complex layer. You have totally 3 layers. Okay, you have totally 3 layers. It is chemically complex. What is the meaning of chemically complex? Because the composition of each layer is different, which is important in a medical point of view. Okay, mechanism of action of drugs. So this is a chemically complex layer from outer, that is from the environment to the inner part of the cell. They are, outermost is the glycocalyx, middle layer is the cell wall, and innermost layer is the plasma membrane. So shall we read the answer, the layers once again? Glycocalyx, the outermost, cell wall, the middle one, and plasma membrane, the innermost. These are the 3 layers of a cell envelope. Now what is the NEET trap here? They can rearrange the layers and ask you the question. Okay, you can see the questions like glycocalyx, plasma membrane, then cell wall, or cell wall, plasma membrane, then glycocalyx. You have to rearrange the orders of the layer correctly. Next, under glycocalyx, if the glycocalyx, that is the outer layer, if it is a loose sheath, you call it as slime layer. If it is thick and tough, you call it as capsule. Now, how many of you are able to remember what I read? Leave the answer in the comment section. Then cell wall, about the importance of cell wall. See, you have seen gram staining. Based on the gram staining, it is divided into gram positive bacteria and gram negative bacteria. That is those which takes up the stain and those which doesn't take the stain. Which one is responsible for that? The cell wall. Out of 3 layers, only cell wall is responsible for picking up the stain. This is also an important NEET MCQ. Next is the plasma membrane. It is the selectively permeable membrane. One similarity with the eukaryotic plasma membrane. Okay, this shows similarity to eukaryotic plasma membrane because lot of differences between a prokaryotic and eukaryotic cell, isn't it? Next, prokaryotic ribosomes are of 70S type. That is 50S is the larger unit and 30S is smaller unit. Here not like max, so don't add and choose an answer 80S. These are Swedberg's units, that is sedimentation coefficient, which is an indirect measure of density and size. Okay. Next you have inclusion bodies. The prokaryotic reserve materials, they are the reserve materials in the prokaryotic organisms. They don't have any membrane, so they will be open, naked in the cytoplasm of the prokaryotic cell. Okay, these are all revision. We can do again the same points many number of times, but we'll finish off once. Then what are the examples of inclusion bodies? Phosphate granules, Synopsis granules, and glycogen granules. The 3 examples are phosphate granules, Synopsis granules, and glycogen granules. Next is the day 4. What we saw is endomembrane system. Now students are able to recollect what are the organelles of endomembrane system. If you know the answer, quickly type it in the comment section. The 4 organelles are endoplasmic reticulum, Golgi, lysosomes, and vacuoles. Do you know why I have written in this order? Because the sequence happens like this only. Okay, the endoplasmic reticulum have the raw materials, that is the proteins. They are the manufacturers. From there it is shifted to Golgi. They are the packages we saw yesterday in the function of Golgi apparatus. Okay, packaging and modification is done here. Then it is dispatched to outside the plasma membrane or to the lysosomes or to the vacuoles. So endomembrane system is made up of endoplasmic reticulum, Golgi, lysosomes and vacuoles. These four are included because they have a coordinated function. Very important MCQ this one. They have a coordinated function. All their functions are coordinated to achieve a goal. Okay, to achieve an action, you can say. But they can give three as one of these three as options. Okay, that is mitochondria, chloroplast and peroxisomes. Students, I wish you to say one thing. I'll reply to the chat only after the session is over. Okay, I am going to concentrate right now here. Once it is over, I'll reply to your chats. But you can post it. So the NEET trap is, don't choose mitochondria or chloroplast or peroxisomes, because the reason is they have independent function. It can be an assertion type of question, assertion reason type of question. Okay, I have mentioned the reason. You should not choose any three of this because they have independent function. They are functioning on their own. No way it is related to all these four. But the confusion why it happens is because of the word membrane. These are membrane bound organelles. So students, you may choose in an exam panic. So make sure that these three are not included in endomembrane system. Now lysosomes. Lysosomes are formed by packaging in the Golgi apparatus. Very important. An organelle which is born from Golgi apparatus. Next is hydrolytic enzymes. The lysosomes contain hydrolytic enzymes which are active at acidic pH. Here they can ask you, give a numerical value like pH of 7 or pH of 11. Which one you will choose? Okay, so this is one NEET trap. If you study like it is active at acidic pH and you go to the exam, suppose if they give the numerical value, you should know the range of acid and alkaline. Okay. Next, nucleus. This is the genetic command center of the cell because it carries the DNA, the blueprint for the cell. Okay. So it is discovered by Robert Brown in 1831. Nucleolus. What is the function of nucleolus? We saw yesterday also, someone told the answer. Now type it in the comment section. It is rRNA synthesis. If you know the answer, type it in the comment section. Nucleoplasm consists of what? Nucleolus and chromatin. It is also called as, what is the other name for nucleoplasm? If you know the answer, type it in the comment section. It is nuclear matrix. If you are following me up, type it in the comment section by leaving your emoji. Okay, next is the block 5, that is the day 5. We saw about vacuole. The important point is tonoplast. It is the single membrane, yes for single membrane covering the vacuole. One membrane which surrounds the vacuole. Okay, covering membrane for the vacuole is called as tonoplast. It occupies 90% of the plant's cell volume. Okay, for example, if it is a plant cell, you have plant cell, vacuole occupies 90% of the cell volume. Example of food vacuole is protista and contractile vacuole is amoeba. What is the function of contractile vacuole? Osmoregulation and excretion. What is the meaning of osmoregulation? I told you already. If you know the answer, type it in the comment section. Osmoregulation means maintaining the water and electrolyte balance of the cell. Water balance and electrolytic balance of the cell is called as osmoregulation. Next you have semi-autonomous organelles. Students, are you able to name the semi-autonomous organelles? If yes, type it in the comment section. What are the two semi-autonomous organelles? Mitochondria and chloroplast. Okay. Similarities. What are the similarities between the two organelles? Both are double membrane bound structures. They have circular DNA, 70S ribosomes. They divide by binary fission. Both are of endosymbiotic origin. Okay. The reason why these two are semi-autonomous organelles is due to the presence of, see, I have written in the same color for identification, that is circular DNA and 70S ribosomes. Okay, because of this two, they can prepare their own needs, meet their own needs. So they are called as semi-autonomous organelles. They prepare their own protein. Here the NEET TRAP is the location of ribosome. For example, there is an assertion sentence which says eukaryotic cells contain only 80S ribosomes. What do you choose? Is it true or false? It is false, because eukaryotic cell contains, the cytoplasm contains 80S ribosomes. Mitochondria and chloroplast contains 70S ribosomes. Both are present. Okay, which means this sentence is wrong. This can be asked as an assertion reason type of question. Okay. Next is, what is the primary function of mitochondria and chloroplast? You know it already. Type the answer in the comment section. Mitochondria, the function of mitochondria is aerobic respiration. Which means what? What is the meaning of aerobic, students? In the presence of oxygen. Okay. Just now we saw an organism which can live in complete absence of oxygen. Name of the organism. Okay, type it in the comment section right now if you know the answer. Okay, next you have chloroplast. The function of chloroplast is photosynthesis. Next, aerobic respiration. What is the meaning of aerobic respiration? That is ATP synthesis. Photosynthesis means sugar synthesis, and aerobic respiration means converting this sugar into ATP. Okay, this sugar prepared is converted into ATP. So ATP synthesis means breakdown of sugar, and it is converted into ATP, which is our energy source. Okay, next you have organelles, the major functions and the primary product. Yesterday we saw this organelles. That's nucleolus. What is the function? Nucleolus, where is it present? Now I am going to tell you, ask you a question. Nucleolus is a membrane bound structure. Is it true or false? Type the answer in the comment section now. Okay, nucleolus means this is nucleus. Inside the nucleus you have spherical structures. There is no membrane. Okay, they are just placed in the nucleoplasm, that is the matrix of the nucleus. So nucleolus or non-membrane bound structure. This is also an important MCQ. Non-membrane bound structure. What is the function? rRNA synthesis. The primary product is production of ribosome subunits. That is, these ribosomes are the ribosomes of the cell. You are studying ribosome organelle, right? Those ribosomes are prepared by this nucleolus. Now you have Golgi apparatus. The function of Golgi apparatus is packaging and modification of materials. I told you from endoplasmic reticulum, the proteins are synthesized and it is sent to Golgi apparatus. Here there is a addition of groups. Okay, so the proteins and lipids are glycosylated to glycoproteins and glycolipids. That is what is the meaning of packaging and modification of materials. So packaging and modification of materials, primary product is glycoproteins and glycolipids. Now ribosomes are the site of protein synthesis. What is the product? Polypeptide chains. These are used for the cell's function. Next you have rough endoplasmic reticulum. Why you call it as rough endoplasmic reticulum? Type the answer in the comment section, students. Rough, R stands for ribosome. Okay, if the endoplasmic reticulum has ribosomes studded like this, they are rough endoplasmic reticulum. What is the function of RER? Protein synthesis and secretion. Secretory proteins. Can you name one secretory protein? Example is insulin. Insulin hormone is a product of the secretory proteins. Okay, rough endoplasmic reticulum. Function is protein synthesis and secretion. The product is secretory protein. Next, you have smooth endoplasmic reticulum. The function is lipid or steroid, that is the hormone synthesis. You have steroid hormones, right? So it is produced by smooth endoplasmic reticulum, lipids and detoxification. Okay, what is the meaning of detoxification? If you know the answer, type it in the comment section. I will make it as a separate shorts. Detoxification is a very interesting topic. Next, lysosomes. Okay, the function of lysosomes is intracellular digestion, that is, it acts as the stomach of the cell. Stomach of the cell. It digests all the waste and biomolecules. Okay, lysosomes are very important. With this, we complete the past 6 days information. Students, hope you enjoyed and you gathered the information. Please leave your feedback in the comment section. It will be helpful for me to further decide what chapter I have to do. If you find any chapter which is very difficult, we can do the same code word reading. You can leave your comment in the comment section. Thank you for listening and watch and following me up. Take care, students. Bye.